Greetings, everyone, and welcome to another exciting genetic engineering and biotechnology news webinar. Our presentation today is entitled Simplifying Protein Quantification Workflows. I'm Jeff Bogaliskis, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Having the ability to quickly and accurately assess protein characteristics is fundamental for the speed with which the biopharmaceutical industry currently operates. While LC-MS generally provides improved multiplexing and specificity in addition to increased accuracy and rationalized method development, its benefits may often be challenging to realize within the world of protein quantification. Yet significant advances have been made not only to expand instrument sensitivity, but also to improve methodologies that provide investigators with simplified sample preparation through streamlining quantification workflows. Selecting a suitable technique to measure valuable protein samples can be challenging for even the most experienced investigator. Let's meet our speaker for today's webinar presentation, who will provide us with valuable insight into overcoming the challenges that are often encouraged when implementing LC-MS approaches for protein quantification. Erin Chambers is a principal scientist for the Waters Corporation. Dr. Chambers will describe her advanced methodology for identifying and quantifying bioanalytical protein samples. So before we get started into Erin's presentation, I just want to remind everyone that we'll have a few interactive poll questions that will show up at several points during the talk. I'll be back on to introduce the questions and allow everyone a few seconds to answer. Just click the appropriate response from the selection on the slide. Additionally, I want to encourage everyone to submit questions for our Q&A session at the end of the presentation. We'll try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. Okay, with all the particulars out of the way, let's get our webinar started. Erin, the floor is yours.
Well, thank you, Jeff, for that great introduction, and welcome, everybody. So as Jeff mentioned, I'm Erin Chambers, and I'd like to talk today about protein quantification using LC-MS via the surrogate peptide approach. And what I'm going to do is I'm really going to focus on some of the practical aspects of this workflow, and we're going to talk a lot about some of the subtleties involved, and ultimately we'll get to ways that we can standardize this very complex workflow. The way I got the talk structured today is that first we'll identify some of the primary challenges and some of the typical steps in this workflow. Then we'll get into antibodies specifically, although these workflows cover antibodies, small proteins, large proteins, both therapeutic proteins and biomarkers. So it's very broad and all-encompassing. Talk a little bit about antibodies specifically, though, and how the choice of signature peptide impacts the workflow. We will get into some of the purification or cleanup steps, both at the protein and peptide level. And then we'll talk about how we can address all of these challenges and how we can really arrive at a very standardized and very reproducible approach to what is essentially a multi-step within multi-step complex workflow. And then finally, I want to wrap up with some work on ADC quantification. So if we look at a protein quantification workflow using LC-MS, we're faced with a number of challenges. So right off the bat, our first challenge is around identifying the unique or signature peptides. And every time I talk about this topic, I'm usually talking about the lab part of it where you're actually in the wet lab prepping the samples, but I always get asked, well, what about identifying the signature peptides? That's the first place we have a problem. So we will cover that briefly today. So then, of course, the next thing is you've got a choice around cleanup at the protein level. So do I want to purify that protein from plasma or other matrices, or do I want to do a direct digestion? And so we'll look at the impact of your various options there and then also talk about how to optimize or how to make a choice based on the ultimate goal of your assay. Next, we get into what is probably one of the most complex steps, and that's the whole digestion step. You can see here that it's actually many steps within a single step. So you've got denaturation, disulfide bond reduction, and alkylation. the digestion with the enzyme, and then there's actually a quenching step as well. And you'll see that each one of these has multiple variables that need to be optimized. So there's a lot of opportunity to make different choices, and for ultimately lack of standardization, which of course impacts reproducibility and the ability to transfer these complex workflows. We will talk about purification of signature peptides and some optimization there. And then we ultimately get into the LC-MS analysis. So I promised that we'd spend just a brief bit of time going through some of the primary challenges. And, I think you may have gotten a sense from the previous slide that there are many, many possible options. At the protein level, you can choose to do no purification. You can do a generic affinity, a specific affinity capture. Some folks, although it's less common, might use molecular weight cutoff filters or depletion plates. And then when you look at the digestion step, you've got a reduction and an alkylation included there. And what we've actually found is that it's perfectly possible to omit those steps and streamline your workflow. But it depends on the protein. It depends on where your signature peptides are. So you see there's lots of questions. And really, as I mentioned, this leads you to that lack of standardization. So many different approaches, different reagents, different qualities of reagents, different modifications to the reagents. Do I pre-cheat? Not. I measure, you measure, they measure in another lab. Do they get different results, different weights? And all this leads to ultimately a different quantitative result. And again, an inability to standardize and to get something that's transferable. And then there's method development. Do I have the expertise in my lab to do the method development? Do I have the time? And when you look at the number of steps and steps within steps that are required to optimize, it becomes a very daunting task. And we mentioned, the reproducibility. And so you're looking at reagents where you've got to consider lot-to-lot reproducibility, things like enzymes. It isn't a given that you've got that reproducibility. Ultimately, we've got to be able to reach very low detection limits. While ligand binding assays have been the gold standard historically, and they are very, very sensitive, use very small volumes, I think folks have become aware of some of the challenges there, which have really led us towards broader implementation and the desire, at least, for broader implementation of LC-MS. And then finally, there's the aforementioned identification of unique signature peptides. So that's one of the places I'm going to start. So just very briefly, I wanted to go through the process. I could honestly spend several hours to a day teaching a course on how you would identify signature peptides and what attributes to look for. I really want to just give you sort of an overview here. So the first thing you want to do is you want to take your protein sequence. You want to do a blast search, which compares it to all the sequences of other proteins in the database. And what it's going to do is it's going to return back those that are most closely related. You will then take that sequence and use a tool called the Sim Alignment tool from Exposi to align your sequence with the most closely related sequence from the database. Now, there will obviously be differences, places where amino acids may be omitted or added, but you want to align all the identical sequences. This then allows you to do a proper in silico digestion, which will generate your potential signature peptides. So lots of tools that can be used to do that. There's one in our very own software, MassLinks and BioLinks. There's also Protein Prospector, Skyline you've probably heard of. It's a very popular tool. Once you have those peptides, you then need to determine which of the peptides are specific. So you perform another blast search, where you take those peptide sequences, blast those against the database, and you will again get a return which will show you how similar they are to other peptides in the database. And ideally, you will want to choose those that are most unique. Now, there are a couple features that make good, unique peptides, a few things that we're looking for or that we want to avoid. Typically, we'd like them to be anywhere from 8 to 24 amino acids. Now, this is typically because the larger the peptide, the less likelihood of an isobaric interference or another peptide with, again, a similar MRM transition. And so as bigger they get, the more specific they get. Now, occasionally we don't have a choice, so we might consider some of the smaller ones. We also would ideally, and again, it isn't always possible, but ideally we'd rule out peptides with known sites for modification, oxidation, deamidation. So we'd be looking for cysteines, methionines, N-terminal glutamic acids, which might cyclize, things of that nature. And ultimately, we'll take those peptides that are on our target list, and we will be looking for the ones that give the best MS response, so the highest sensitivity. We'd be looking for those that retain well chromatographically, have good peak shape. And then, of course, we would monitor those in a matrix so that we could, again, look for specificity. Now, when we talk about purification or cleanup, we talk about it, again, at the protein level and the peptide level. And I just want to give you a little sense here of what the different types of purification at the protein level look like. And ultimately, you're going to have to make the decision as to which one fits your assay need. So if we look all the way over on the right there, this is a direct digestion of human plasma. And what I want you to notice is the background. On the bottom, that is our lowest level sample, all the way up to the highest on the top. So if you look at the no cleanup sample, the direct digestion, you see a lot of other endogenous peaks, even though this is an MRM, or an extracted ion chromatogram of a very specific transition. You can see that there are a lot of other extraneous peaks. This was done on a triple quadrupole, so remember it's a nominal mass instrument. However, that's a very common choice for this type of work, very, very sensitive. Now, if you look at the middle panel, if we do a generic affinity purification, so this might be something like a protein A or a protein G in human plasma, which will pull out the IgG1 subclass from that plasma, you see that we reduce the background to some degree. Not completely, because we've still got roughly 15 milligrams per milliliter of protein present in the sample. So it isn't quite as clean as we'd like it to be, but we've improved it somewhat. Now, where we really are able to drive detection limits down is when we look at a very specific capture. So in this case, it is a kappa anti-human purification in rat. So it acts as a very specific capture here. And you see we only have one peak, and that is the analyte of interest. So working down the workflow, that's purification at the protein level. And of course, what you ultimately decide will be based on the sensitivity and specificity you need. Next, we take a look at the digestion step. And I alluded to this previously, the fact that there are up to 5 steps: denaturation, reduction, alkylation, digestion, and quenching. And each one of those steps has multiple aspects that must be optimized. You're going to look for the specific enzyme, the vendor, the ratio, the temperature, the time for many of these steps. You may even think again about omitting some of the steps, but how do I know which one? So ultimately, you can really start to see how this leads to a complete and utter lack of standardization, and certainly to a question about the ability to reproduce these complex methods lab to lab. Now, a little bit more detail here on the digestion step. This first panel just shows you the impact of increasing digestion time. So you increase the efficiency up to a certain point, after which the efficiency stabilizes. And then if you took this out far enough, you'd start to see autocatalysis, whereby you've got reduced efficiency. But what amount of time is it? And the amount of time will depend on the temperature. It will depend on the specific reagent. So you've got a lot of things to think about. You also have to couple that with the ratio of enzyme to substrate. And here again, too little is not as optimally efficient, nor is too much. So there's a happy medium there that one has to figure out. And then finally, of course, let's use an example of a trypsin, for example. Not all trypsins are created equally. Some have been modified to certain types of stability or reduced activity. Others from different vendors, different purities. And what you're looking at here is sort of the efficiency of both a signature peptide and its labeled counterpart. And so you can certainly see that different trypsins from the same vendor, from different vendors, not only do they have very, very different costs, on the order of 3 orders of magnitude different in some cases, but also very different performance. And this is just 9 of them. There are many more than that to consider. When we look at reducing agent, again, need to think about concentration. And here I'm looking at both a unique surrogate and a generic framework peptide from an antibody. And what you can see here is that as I decrease the reduction agent concentration, I've hit an optimal efficiency, for example, for the generic peptide, which is different from the unique. So where do I draw the line? How do I decide? Furthermore, different possible reducing agents have different optimal temperatures, different optimal concentrations. So just within the realm of reducing that sample, there are 4 variables to optimize. So again, this requires quite a bit of time from your scientists to do that. The step we want to talk a little bit about is the peptide purification. And some folks may ask, well, why do I need to do that? I'd like to just inject my sample directly. Well, this is a perfect example here of why you might want to purify those peptides. So in addition to removing salts that could reduce MS uptime, in this example here, when I look at a pure peptide up on the top panel versus on the bottom panel, that same peptide in the presence of serum digest matrix, I notice 2 things. Our retention time has shifted, and I've lost about 2/3 of the area counts. So this can be compensated for or improved through the use of peptide purification. But what method? And do I have a method that might be generic for all tryptic peptides, for example? So before we get into the next section, I think it's time for a few poll questions.
All right, everyone, here's our first 2 polling questions. Our first one asks, what is your biggest challenge to using LC-MS for bioanalysis of large molecules? Select the best answer from the list below. I'll give everyone a few seconds. All right, let's move on to our next polling question, which is, what would you like to improve about your workflow? Again, select the most appropriate answer from the list below. All right, let's continue on with Erin's presentation. Erin?
So now it's time to talk a little bit about antibodies and how the choice of peptide can impact the methodology and the steps that we include or omit. So we look at a few examples here, and I'm actually going to very briefly show you 4 examples just so that you start to see some of the commonalities. So the first is Infliximab or Remicade. And for each one of these, you will see that I've highlighted in a red box the unique signature peptides. These are unique specifically to this monoclonal antibody. In the blue boxes, you will always see the same peptides highlighted. These are framework peptides from the conserved region, so they will always be the same. So if we look at our next example here, which is Trastuzumab or Herceptin, you'll see again we've got 2 very unique peptides in red, and you'll also start to see here that those unique peptides tend to sit out on the fringes, out in the variable regions. So that actually makes them a little bit more accessible, and that's a hint to what I'm going to get to in a moment. Our next one is bevacizumab, and you see the same sort of thing where we've got these unique peptides in red and then those same framework peptides in blue. When I get to adalimumab or Humira, again, sitting in the variable region are my unique signature peptides, and then the same framework peptides. So you ask yourself possibly, why is this important? And the reason is that we want to consider whether or not we can eliminate reduction in alkylation. This can actually save us quite a bit of time in the overall workflow. So looking at those unique signature peptides out in the variable regions there, I've looked at the efficiency of a 3-step protocol where I've eliminated reduction in alkylation compared to the full 5-step. And what the data shows you here is that signature peptides, unique ones, from 4 monoclonal antibodies, we find that the 3-step or abbreviated protocol actually works just as well as the 5-step, if not significantly better. So this tells me that it's very likely I can use this abbreviated protocol. Conversely, when I look at those peptides that are buried deep within the conserved region, more often than not, I actually need that reduction in alkylation step in order to optimize the area counts. So a little bit of a quick background. So now I'd like to talk about purification for just a moment before we get into some of the data based on a solution aimed at standardizing this whole process. So when we look at, so I do want to take just a moment to show you the impact of pellet digestion. So if we digest a sample with albumin and with trastuzumab present, you can see that the albumin peptides elute exactly in that gradient region where all of my unique peptides would elute. So this has the potential to cause coelutions or suppression. And the fact that albumin is present at very, very high concentrations in the sample, on the order of, say, 50 to 60 mg per mil, that also means I need to use significantly more of a reagent like trypsin. Now, the impact of doing a simple protein precipitation pretreatment can be very dramatic. If you look over on the left, this is our simple direct digestion. And what I'm looking at are the area counts for a unique peptide from Herceptin in blue and the area counts from albumin in red. And by using different reagents in different ratios, I can essentially tune into a point where I can eliminate the vast majority of albumin while increasing the area counts for my unique peptide. And in this particular case, this is only a representative of some of the data that we have generated looking at this phenomenon. In this particular case, solvent 4 in a 1-to-1 ratio eliminates a little over 60% of the albumin while more than doubling the area counts for the Herceptin peptide. And there are other reagents that can easily eliminate 80 to 90% of albumin. So you see it can be quite effective. Now next I want to go back to this slide around purification at the protein level. And this time I want to show you what the impact can be on our detection limits. So using a very specific capture, you can see here that my 50 nanogram per mil sample, that's 2 up from the bottom on the far left, I can easily exceed that 5x the level in the blank that's required for an LLOQ, and the signal to noise here is exceptional. So typically, I can expect with a specific capture limits of quantification that are well below the 50 nanogram per mil level. Doing a generic capture, what we've observed in our studies is that we're getting detection limits, not quantification limits, in the range of 50 to 100 nanogram per mil, so a little bit higher there, and in some cases significantly higher. And when I go to a direct digestion of human plasma, it's very difficult to get a detection limit much below 100 nanogram per mil. So we're often looking at quantification limits in the several hundred nanogram per mil to microgram per mil or so. So everything we've talked about so far leads us to think about, wow, with all this complexity, what would it take? What do I need to be able to standardize this? There are a lot of things that I need some sort of standardized approach to be able to do. I need it to accommodate lots of different pretreatments, from protein precipitation to affinity capture to direct digestion. I need some kind of generic method to be able to accommodate the omission of certain steps or the inclusion. It needs to accommodate a range of sample volumes, from small and preclinical tox species all the way up to larger volumes from human samples, and if those volumes are needed to increase sensitivity. It's got to work for small proteins, large proteins, ADCs, antibodies, and everything in between. Naturally, it's got to be able to give me the sensitivity I need. And what I'm really looking for here is reproducibility and accuracy. Now, I think we're right up against one more poll question here before we get into some very interesting data.
Hello again, everyone. It's time for another poll question. The question is, what is your biggest challenge to deploying LC-MS for large molecule bioanalytic work in your lab? Select the most answer from below. All right, back to Erin.
So in my lab, we've actually spent the last few years, believe it or not, researching all the subtleties of protein quantification to ultimately end up with something that we can put forth as a potential solution to all of this complexity and to all of the issues that folks face with transferability and standardization. So I'd like to go over some of the key attributes that came out of this research and ultimately are represented in what is commercialized as something called the ProteinWorks Express Digest Kits. So what's important to note about these? For small proteins in plasma, you're looking at a 10-minute to a 30-minute digestion. When you're looking at antibodies or ADCs in plasma, 3-step protocol, it's about 2 hours. And when you're looking at the total 5-step protocol, about 3 hours. So what you see here is that with these abbreviated digestion times, you're actually able to get these samples completely processed and ready by lunchtime versus the old historic way of waiting 6 hours to overnight. We've fully optimized every aspect of that digestion to be able to bring it down to this level of efficiency. We've got completely standardized protocols. It's essentially a recipe. Format that it comes in allows you to do a couple samples at a time, up to a 96-well plate all at once. And the aim here with all of this, again, is to standardize to reduce variability and to give you reproducibility, accuracy, and precision. We're also going to show you how sensitive this can be. And you'll start to see that this has essentially eliminated method development for discovery-type studies or for those with limited proficiency in complicated protein quantification workflows. There's no capital investments. It takes advantage of the equipment that you have. It works for ADCs as well as small biomarkers, medium-sized proteins, and it even allows you to detect conjugated peptides from ADCs. For the initial set of data that we'll look at, we'll focus on some of the important monoclonal antibody-based drugs that either came off patent recently or will do in the next couple of years. So we're going to focus there initially, and then we'll talk about some biomarkers, smaller proteins, and we'll wrap up with some ADC work. Now, one of the most important things about these complicated workflows is getting reproducibility, accuracy, and precision. And here we looked at multiple peptides from multiple antibodies simultaneously across multiple kits. And this represents the intra-kit variability or reproducibility. Now, the average CV here is actually right around 3%, which is actually pretty remarkable. When we look at the inter-kit reproducibility, the CV is about 6%. Now, in both cases here, these are raw area counts. So that really tells you the power of the precision that's achievable with this approach. Furthermore, this was done with 5 different lots of trypsin, one of the critical reagents, 2 different days of preparation, different MS systems, and different analysts. And so as I mentioned, you've got that about 6% CV for inter-kit and about 3% for within-kit, which is pretty remarkable, but really that is what we're looking for from a workflow like this that we'd like to be able to transfer across labs and across analysts and always get the same result. But when it comes to peptide purification, you recall this slide probably from earlier where we've got a big problem that we've got to deal with in terms of maximizing our area counts, eliminating background, and ultimately increasing uptime from the instrumentation. So the solution to these challenges at the peptide level is to do a purification. And we've chosen to do this with a mixed mode SPE sorbent. So the sorbent has both reverse phase and cation exchange. Now we've chosen cation exchange for this purpose not only because of its specificity, but also because it is well matched to the tryptic peptides. And as you recall, probably they cleave at lysine and arginine, giving nice basic handles there for a strong cation exchange. Now what we've done is we've devised a single generic protocol that is aimed very, very specifically around purification of tryptic peptides. So we've optimized both the wash and the elution solvents to create a nice little window around those tryptic peptides. And what you can see here is that for a variety of peptides, both unique and generic arising from monoclonal antibody-based drugs, as well as from a generic antibody-based internal standard and a smaller protein, we're always getting greater than 80% recovery. So we can simultaneously recover these while eliminating salts, phospholipids, excess digest reagents, and the like. Not only that, but because of the format we use here, we can also reconcentrate the sample. So once you add all the digest reagents to your initial plasma or other matrix sample, you start to bring up that volume. So with this device format, we can reduce that back down to 25 microliters with the elution. So let's now take a look at some of the data that we've generated using this standardized kitted. The first piece of data that I'd like to look at is around Infliximab. So here we've managed to get excellent linearity and accuracy and precision for all the standard curves from multiple different peptides, from different ranges from 50 nanograms to 100 nanograms, all the way up to multiple hundred microgram per mil. In addition, I'd like to draw your attention here to the precision. So this is something that we really care about. What you're looking at here are all the QC samples from the aforementioned study. And all the way from the low QC to the highest QC, you can see that the average precision there is in the low single digits. So this is actually pretty remarkable when you consider the overall complexity of a workflow such as this. And in this particular case, we've used a generic protein A affinity purification up front, and we've then taken it through the ProteinWorks kit for the rest of the workflow. So that entire process, and we've got single-digit precision. Furthermore, we also have the required sensitivity. And so if you look at what I'm showing you here, we're able to get down to 10 nanograms per mil for Infliximab from protein A, followed by the ProteinWorks standardized kit-based approach. So to get this type of accuracy, precision, and sensitivity from a kit and a completely generic standardized approach is pretty remarkable. Let's now take a look at some other data from other antibodies. This is Trastuzumab. This is actually a whole plasma digest. And the dynamic range there is 100 nanogram per mil for our limit of quantification, limit of detection. It's right around 50. And again, our accuracy is typically in this single-digit range. Moving on to yet another antibody. When I look at bevacizumab or Avastin, look at my QC statistics, multiple signature peptides, and again, I'm looking at very, very low single-digit precision there and single-digit accuracy from a completely generic standardized approach. But remember, again, this is a kit, so we hope and we are now demonstrating that that's the type of performance you can get. The final antibody-based example is for Humira or adalimumab, where we're able to get not only the linearity and the accuracy and precision, that data is not shown here, but also the quantification limits that are expected from this protein. Now, another thing that it's important to have from a standardized approach is the ability to deal equally well with different starting plasma volumes. Now, if you're familiar at all with quantification based on the signature peptide approach, you're probably aware that typically one needs to actually do a lot of calculations up front based on the total protein expected in the sample. And depending on the starting volume of plasma, that total protein changes, also depending on the type of pretreatment. So we've taken all of that work away. We've grossly simplified the approach. We've done all of the research and able to provide you the protocols that accommodate all of this. And what I'm showing you here is that we've looked at, this is just one example, 4 different antibody-based drugs, 2 to 3 different signature peptides for each drug, simultaneous quantification, and you get excellent performance, whether it's 15 microliters that you're starting with or 70 microliters that you're starting with, in terms not only of linearity, just looking at a couple points here, but also for the accuracy, with average accuracy right around the single digits. So it accommodates a wide range of starting plasma volumes. And in case anybody was interested, this was all done as a simultaneous quantification of the 4 antibodies. So we've talked a lot about monoclonal antibody-based drugs. I did want to get into a few biomarkers here. And the first that we're going to talk about is Cytochrome C, using 35 microliters a sample. It is a smaller protein, about 12 kilodaltons. This required only 10 minutes to digest, so a very, very fast digestion. Linear fit for the 4 signature peptides we show here, highly accurate and precise with single-digit accuracy and precision for both. When we look for Cytochrome C at the QC samples, again, this is where the precision becomes pretty remarkable. It's in the low single digits. The average is right around 2% or so. And you consider, again, this is a highly complex workflow. It's a simple 10-minute digestion, very accurate, very precise, which is exactly what we're looking for. If we want to see what the chromatography looks like for one of the signature peptides, this is a series of QC samples. You can see at 3 micrograms per mil, we've got more than adequate signal to noise. And actually, our detection limit is well below that sample that's shown here. And we're actually in the nanogram per mil range for detection, which again is something that we like to have for the assay. I want to move along now to another protein of interest, apolipoprotein A1, or ApoA1, as folks know it. It's a little bit larger protein, about 28 kilodaltons. In this case, we're starting with only 15 microliters. So ApoA1, as you are probably aware if you work with this protein, is present at pretty high levels. A typical circulating range is around 0.9 to 1.3 mg per mil. And reductions in that are what folks are concerned about. So we did a quick time course, and what you can see here is that optimal digestion time was right around 15 minutes. So only 15 minutes to digest this protein. What does some of the data look like? Looking at my standard curve range from 10 micrograms all the way up to 1.5 mg per mil. Nice, simple 1 over x squared weighting, good linearity, excellent accuracy. And if I look at the bottom there, down at the QC samples, you can see again the precision in the low single digits. Quick 15-minute digestion, only 15 microliters of sample. And this is just an example of the representative chromatography in rat plasma. We did these experiments in both rat and human. Human will obviously have some endogenous levels. Rat should not have any, as the sequence is different in rat, and that's sort of what you can see here to highlight the chromatography. When we do go on to look at human plasma QC samples, we calculated the average endogenous level across multiple pooled lots to be roughly about 600 microgram per mil. So this 600 micrograms is a little bit lower than the reported range, but it is consistent with the findings of Pasella et al., who discovered that if you store the plasma at 4 degrees for over 2 weeks, that you actually start to see reduction in the level. And in fact, the plasma pools that we used for this sample were stored for well over that time period. So we did see the commensurate reduction here. But what's important, if you look at the human samples here, and these were quantified using the standard addition level, that again, you look at the CVs in the low single digits and the accuracy again in the single digits with all QC samples passing. Now, one final example of interest here is CRP, or human C-reactive protein. Again, not a huge protein, about 25 kilodalton, but a very important clinical marker for inflammation, infection, and atherosclerosis. We started here with only 35 microliters of sample. Again, we don't need a lot of sample. The quick 3-step digestion was more than adequate, and I'll show you in a moment where we ended up with that time, a very, very short digestion time as well. Quick chromatography of the 3 signature peptides that we identified from CRP, and here we used a silica-based, high-strength silica-based column for retention. And what we decided to do once we got to CRP was to go back and sanity check everything that we did for the kit. So we went through and we looked at a lot of data around antibody-based proteins, some of the smaller proteins, and every time we used the standardized kitted methodology. So here we did a bit of a sanity check for ourselves. So the first thing was to sanity check the denaturation. And we're looking here at multiple denaturation times, and what we found was that we confirmed exactly what we recommended in the kit for denaturation. Looking at digestion time, you can see here that by and large, for multiple different peptides, we maxed out right around by the half-hour time point. Now, once we got up over 2 hours, up to the almost 3 hours, we got a little bit more for 2 of the peptides, about 20% more. But if I had a choice between half hour or 3 hours, getting 20% more, I'm likely to stick with that half hour. So again, confirming that we could use these short time frames for digestion for some of these smaller proteins. And then finally, the quench time, we recommend in the kit that 5 minutes, and that's exactly what we confirmed. So it was really just a sanity check there, which confirmed, again, all the recommended kit conditions. We pulled that sanity check one step further to the recommended SPE, and again confirmed here that the recommended condition of eluting with 40% organic indeed held true not only for all of the aforementioned antibodies, the ApoA1, the Cytochrome C, but also for CRP. When we finally look at the chromatography settling on a 1-hour digestion, we calculated out the endogenous concentrations. So you see the human plasma unspiked blank down in the bottom left, and we got average plasma concentrations very close to 3 microgram per mil, which was well within the reported circulating plasma range for CRP levels of 0.75 to 15 micrograms per mil. Now, I'll take a moment just to look at how the competitive products stack up here. And what we found is that whether you use the 3 or the 5-step Waters protocol with the Waters kit, we get significantly higher area counts for unique signature peptides as compared to the most commonly used competitive kit. The magnitude of the difference here becomes even greater when we start to look at those framework peptides that are deep within the conserved region, whereby this kit provides area counts that are anywhere from 2 to 5x higher relative to the competition. Another point I want to draw your attention to quite quickly is the use of a generic antibody internal standard. We, in fact, used a murine antibody not only as a generic internal standard, but also as a workflow proficiency testing standard. You can see here on the top the chromatography from the signature peptides for this generic internal standard. And what we did was essentially choose the closest eluting peptide to that signature peptide from our target protein. And all the work that you saw previously, the overwhelming majority of that was done using this generic antibody internal standard, except in the case of some of the smaller proteins whereby we used an actual labeled peptide. So before I get into the final segment, which is looking at ADCs, I want to pause for a moment just to do a final poll question here, and then we'll talk about ADCs.
Hello again, and it's time for our last poll question of the webinar. We would like to know, how important is automation for LC-MS protein quantification workflows? Please select the best answer from below. All right, Erin, continue on with the last part of your presentation.
Okay, great. Well, I'd like to thank you all very much for taking the time to answer some of these poll questions here. I think it always helps not only us, but ultimately, a little bit of information for yourself as well around what's happening in the industry. So now I'd like to go ahead and talk about ADCs. In particular, I'd like to talk about total antibody quantification for Trastuzumab emtansine, or TDM1 as it's known. Now, this is a lysine conjugated ADC. There are 90 possible sites of conjugation. And one of the other labs that I work with here at Waters did all of the site occupancy testing, some of the identification and relative quantification of conjugated peptides. And what I will focus on here is total antibody quantification using the ProteinWorks Express Digest kit. Now, one thing I want to mention up front there is that any site where you've got conjugation to that lysine makes it more difficult for the trypsin to cleave. So we do need to think a little bit about this when we choose the signature peptides that we're going to use for quantification. We need to consider whether they are arginine-arginine at either end or whether there's a lysine present there. And we need to couple that to anything we know about site occupancy. So the way that this study was designed is that we created standard curves using both Trastuzumab, or the unconjugated antibody, as well as the ADC TDM1. And then we used each of these curves to quantify both its own QCs as well as the QCs of its complement. So the data we're going to be looking at is using the Trastuzumab standard curve to quantify Trastuzumab ADCs, as well Trastuzumab QCs, as well as the ADC or TDM1 QC samples. Let's look briefly here at our choices for unique peptides in red. So 2 out of the 3 actually cleave at a lysine. One of them, the IYPT, cleaves on arginine-arginine. So the thing we have to consider here is the site occupancy at the lysines. Now, looking back at the data that was generated by my colleagues for those 2 unique peptides there, the site occupancy was very low, on the order of 1 to 5%. So if anything, we might expect only a very slight negative bias in the data for those 2 peptides. So looking at the ADC standard curve statistics for the 3 possible peptides here, including in this case one of the generic framework peptides, you can see that across the expected range from about 100 nanogram per mil up to 500 micrograms, we use a very, very simple 1/x weighting, excellent accuracy, and a mean accuracy of all the curve points right around 100%. Now I want to take each one of these peptides on its own and look at the data for the QC samples. In this first case, if you focus on the bottom table, those are all the QCs for the ADC. The top table are the QCs for Trastuzumab. In either case, you're going to see here that the precision for all of the CVs, again, is in the low single digits as well as single-digit accuracy. So you see here that we can use this standardized kit-based approach to get high accuracy and precision for total antibody quantification for TDM1. That's just one peptide. Now look at the other 2 peptides, again focusing on the bottom half of the table here, which is the ADC. You see again, excellent accuracy and precision. And then the third peptide here, single digit, the average there is right around 4% precision for the peptide, all the peptides, in fact, arising from the ADC TDM1. So that's exactly what we like to see. Now, the other thing that's very, very interesting that we can do with this kit, we can actually observe and, in fact, quantify the missed cleavages. So these are our conjugated TDM1 peptides. Now, you consider here that these are actually going to be in very, very low concentration. So concentration reported there on the bottom is the concentration of the total ADC. When you take into account site occupancy at this particular site, the concentration, in effect, of this conjugated peptide is really only 1 to 5% of that which you see here. So you see we're looking at two different conjugated peptides on the left and the right, and you see the telltale diastereomer pair, and also the fact that we're looking at this fragment at 547, which is diagnostic or representative of the hydrophobic small molecule payload. Furthermore, we can confirm the presence of these conjugated peptides by the fact that we've got multiple different transitions, both that 547 and the 485, that both show the presence of the nice diastereomer pair. Furthermore, we can confirm this by the increasing concentration of these conjugated peptides that occurs with increasing ADC concentration. So the signal is concentration dependent. Now, when we look at how the ProteinWorks kit compares to competitive kits for ADC total antibody quantification, as well as you'll see in a moment for the conjugated peptide determination, you'll see some significant differences. Here, we're looking at the signature peptides for total antibody quantification. And again, you can see in all cases that the Waters kit will give either significantly higher area counts or at least comparable. Now, when we look at the background on the IYPT peptide, you can see we can actually only quantify this using the ProteinWorks kit. Looking at the competitor on the right there, the level in the blank for the interference is so great that we can't even quantify this peptide using the competitive kit. Another thing that's important here is when we look at conjugated peptides. So you see on the left we can visualize these and, in fact, quantify them very nicely with the ProteinWorks kit. And, in fact, with the competitive kit on the right, even up at 250 micrograms per mil, we can't even really see an accurate representation of that diastereomic pair. So I'd like to wrap up, and I think you've gotten an appreciation, if you didn't already have it, for the complexity of these workflows. The diversity of the steps, the numerous nature of the steps, the subtleties of the steps within the steps, all of which you would have to optimize. And now what we've done is we've taken our years of research and put them into this kit, this standardized generic kit, which you can see, and hopefully you've gotten appreciation from the data, which delivers single-digit accuracy and precision, not only for numerous monoclonal antibody-based drugs, but also for total antibody quantification for ADCs. It allows us to quantify even their conjugated peptides, and we're able to quantify a vast array of biomarker proteins as well. So it's truly broadly applicable and truly delivers sort of on that promise and on that desire for standardization and reproducibility, which ultimately allows us not only to get the same results within our own labs from analyst to analyst, day to day, but also allows us to transfer and, to have that ultimate confidence in our results. The last thing I want to do really is to acknowledge the other folks, not only in my lab here at Waters, but also in the other labs at Waters that have contributed to this work. I'd like to wrap up and turn back over to Jeff, and I think we probably have a few minutes for questions. So thank you all very much for attending today, and hopefully we've got a couple good questions coming our way.
Thanks, Aaron. That was a great presentation. I think you provided our audience with some great information about new techniques and products from Waters that will aid many investigators' bioanalytical workflows. Before we start the Q&A session, I want to remind everyone again that this is your final chance to submit questions for Aaron. So hurry up and submit them now. All right. So why don't we get started with the Q&A session. I see we already have a few really great questions that came in, so we'll try to get to as many of them as possible. Just bear with us for a few seconds while we get everything set and ready to roll for the Q&A. All right, Aaron, so our first set of questions are here. The first question from one of our audience members asks, how does your kit differ from other kits currently on the market?
Okay, Jeff, thanks for that question. That's a good question. There are a few other options out there. I think what I'd say here is that we spent a couple of years designing this specifically to be not only very generic and broadly deployable, but also to have the appropriate degree of flexibility to accommodate everyone's needs. Now, a few things that are specifically worthy of mention are the fact that there are two different versions of a kit, depending on whether you're doing an affinity purification up front or a direct digestion. Now, that is unique, and that allows us to really optimize the sensitivity in both cases. If you've been doing this work, you know that a lot of the efficiency is dependent around the protein content. So, designing kits that are specifically geared towards these various instances allows us to provide a lot more sensitivity and a lot more optimization. So that's one aspect that makes it very unique. In addition, it was also designed to provide optimal performance not only for those peptides that are out, let's say in the case of monoclonal antibodies out there in the variable regions, but also for those unique signature peptides that are buried deep within the conserved regions. So, this really allows for that optimal performance, highest sensitivity for a broad variety of different signature peptides. And when you're first working on identifying a signature peptide, it doesn't matter if it's from a monoclonal antibody or a smaller biomarker protein. You don't know where that peptide is going to exist nor what its attributes are going to be. So to have that type of flexibility and a kit that's specifically designed to provide optimal sensitivity is very important. A few other critical attributes here are that the digestion time is very fast, anywhere from a couple minutes up to 2 hours for an ADC or an antibody. That leads me to another very important distinction. There's a lot of interest in ADCs. And I think you could see here that not only can we do total antibody quantification, but we are very unique in that we can observe and quantify conjugated peptides, which can be at very, very low abundance depending on site occupancy. So that's very unique. And then the last thing I'd say is that it's flexible to accommodate very small volumes up to larger volumes, so dealing with preclinical species, rodent species, all the way up to human. So, that was an excellent question, and I think that's probably how I'd answer that. Thanks, Jeff.
Great. Thanks, Aaron. It was a very detailed explanation, so we thank you for that. The next question starts off by saying some people use filter plates or reverse phase SPE for peptide purification. Why did you choose cation exchange SPE, and how does it differ or impact peptide purification when compared to these other approaches?
Okay, great. Yeah, that's also an interesting question. Certainly an easy approach is to use filter plates, but you don't tend to remove all of the things that we want to remove if you use a more specific approach. So, you've got all these excess digest reagents, salts in there, phospholipids. These can all significantly impact not only the robustness of the assay in terms of instrument uptime, but also the specificity, the sensitivity. You run the severe risk of coelution. So filter plates, reverse phase only SPE, These are not very specific ways of purifying that tryptic peptide fraction. So we chose instead to use a mixed mode cation exchanger sorbent where we're able to eliminate things like phospholipids through the reverse phase portion of the sorbent, and then also eliminate all those salts, excess digest reagents, and we bind the tryptic peptide fraction very, very specifically by cation exchange, which is a very good match for tryptic peptides, which have, again, that handle on the lysines and arginines. So what this gives you is specificity, and that's first and foremost. It also eliminates all of the, the lipids, the salts, excess digest reagents, buffers, etc. But it also, equally as importantly, provides orthogonality. So if you remember, you're doing your chromatography, your LC chromatography there in the reverse phase. This provides that added orthogonal dimension of specificity and purification there.
All right, thank you, Erin. Next question. One of our audience members would like to know, how much do differences in MS instrumentation influence sensitivity of the kit protocol analysis?
So the kit itself will work the same no matter what in terms of the the reproducibility of it, the accuracy and precision. The sensitivity, however, is and can be dramatically influenced by the MS instrumentation. Now, I will also say that LC chromatography is equally at play here. We used a very low system volume, low dispersion system for the LC. We also rely on sub-2 micron particles, very specific column there based on the acuity particle, and a column that's very specifically designed for peptide analysis. It's even QC'd for peptides. So, everything has to come together to make this work. On the MS side specifically, I think what you'll find in a lot of these traditional bioanalytical labs are MS systems that were initially designed for very, very high sensitivity of small molecules. So your typical instrumentation in these labs are going to be a lot of SciEx instruments. These were very specifically designed for high sensitivity of small molecules, and they do that quite well. Here we're talking about large molecules. So the need on the MS side is very, very specific. It helps to have a broad dynamic range so that you can accommodate some of those larger tryptic peptides that may arise from larger proteins. In some cases, people are even using other enzymes which form even larger peptides, signature peptides. So the mass range is important. Another thing that people don't often realize is that even if you have the mass range on some of these other instruments, most of them don't have the sensitivity required up at the higher M over Zs. Now, the instrument we used here, which is a Xevo TQS triple quadrupole, is unique in that it has very, very high sensitivity, especially above M over Z 900. So going up above there into the 1,000, 1,100, 1,200, 1,300. So this can be very, very important. Anytime you're doing any type of peptide analysis. It could be a therapeutic peptide or a signature peptide for protein quantification. So the MS system here is equally as important.
All righty, thank you, Erin. Our next question starts off by saying, you reference using this kit for monoclonal antibodies and specifically for therapeutic proteins in your presentation. Does this kit work for proteins other than monoclonal antibodies and for proteins other than therapeutics, protein biomarkers, for instance?
Okay, right. So that's also a good question. It is true we focused a lot on a lot of the biotherapeutics coming off patent, these monoclonal antibody-based drugs. But I think, there was a lot of data in there that showed its applicability to smaller proteins and specifically to some of the biomarkers like apolipoprotein A1. We also showed Cytochrome C, C-reactive protein. So we continue to do a lot of work in that area as well. So our aim is to start to become more encompassing with that biomarker space as well. And you've seen the beginning of that with the ApoA1 data, the CRP data, and the Cytochrome C that shows that the kit is fully flexible not only for therapeutics, but also endogenous proteins, and that it doesn't even matter what size they are. They could be very small, something like the Cytochrome C we showed around at 12 kilodalton, all the way up to monoclonal antibody range and even larger.
All righty, Aaron. Thank you. And it looks like with this one, this might be our last question. One of our audience members would like to know, they say, I noticed that you've shown data with single-digit percent coefficient of variation for precision and accuracy. Was that data generated using an internal standard?
Okay, so that's a good question because actually there's a lot of data in there when we show standard curves, QC samples. That, of course, was done using an internal standard, and typically we've used that generic mouse antibody internal standard. So with that, it is pretty remarkable, and you know, I've been in bioanalysis for well over 22 years, and it's, you know, I typically see CVs in the 7 to 8% range, and for a workflow as complicated as this one is, to see those 2 to 3% CVs I thought was pretty remarkable, and you know, I think we're really proud to see that because we did put several years of research into this. So a lot of that data, the QCs, the standard curves, all that, was definitely done using an internal standard. However, when you look at the inter and intra-kit CVs, and those were 3 and 6% there, those are on raw area counts. So we did not even have the benefit of an internal standard. Those are pure raw area counts off the signature peptides. So that's even more telling in terms of the ability of a kit like this to fully standardize this very complicated multi-step protocol and to provide the reproducibility that, you know, we thought was really unattainable. So yeah, that data in particular is fairly remarkable when you look at the fact that that was generated on raw, unadjusted area counts.
All righty. Thank you, Aaron. And with that, we've come to the end of our webinar. I want to remind everyone that the webinar will be archived on our site at www.genengnews.com for 1 year. So if you missed parts of it, you can catch it again, and feel free to forward it to the link to your friends and colleagues, which we always recommend. I'd like to thank Erin again for her informative presentation, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Waters for sponsoring this webinar. Hopefully we'll see you again in the future for another Gen webinar. Goodbye for now.
Thank you, Jeff.